So HPL seminar today features the director of Human Performance Lab, Dr. Walter Herzog. Dr. Herzog did his undergraduate training in physical education at the Federal Technical Institute in Zurich, Switzerland, and then completed his doctoral research in biomechanics at the University of Iowa in 1985. It was in this year that Walter started his academic career here at the University of Calgary. And currently, Dr. Herzog is a professor of biomechanics with appointments in kinesiology, medicine, engineering, and vet medicine. He holds the Canada Research Chair for Cellular and Molecular Biomechanics, and he's appointed the Kila Memorial Chair for Interdisciplinary Research at the University of Calgary. Now, as most of you know, Walter is interested in musculoskeletal biomechanics, with emphasis on the mechanism of muscle contraction at molecular level, and the biomechanics of joints, with focus on mechanisms of onset and progression of osteoarthritis. His contribution in the field of biomechanics is unquestionable. He is the past president of the International, American, and Canadian Societies of Biomechanics, and was recently inducted into the Royal Society of Canada. 2017 is a special year for Walter, as he received the Malbridge Award from the International Society of Biomechanics at Brisbane for his career achievement in biomechanics. The award is named after Edward Malbridge, who was the first to use cinematography for the study of human and animal movement in the 19th century. The Malbridge Award is the most prestigious award of the Society, and one of the most notable names in the recipient list is Dr. Ben O'Nick, who is not here, unfortunately. Now, I was lucky enough to be among the audience at Brisbane, when Walter delivered his award lecture for the first time. And in his talk, he mentioned that it was hard for him to talk about the greatest impact he has in science, because this is typically done by someone else, not by himself. However, he does take pride in the education of his students. Indeed, to date, Walter has more than 100 trainees under his name. 68 of them are graduate students and 45 postdocs. When talk about science, Walter is never short of passion, and this passion is always contagious. And I happen to be one of the lucky ones to have the honor to learn from Walter, and I can tell you that Walter is the best mentor I ever have. Now, today I'm excited that we have the chance to listen to Walter delivering his Malbridge Award lecture for the second time. The title of his lecture is Reflection on Muscle, of Intuition, Truth, Serendipity, and Paradigms. Please, Walter.
Thank you very much, Anquan. That's a very, very kind introduction. So this is, as Anquan mentioned, the Malbridge lecture, and our Dean Penny Werthner asked me to give this, and I refused about twice in a row, because I think a lecture like this one can only be given once. You're invited to give it. It's a special occasion, a special place, a special this, a special that, and I think it's very, very hard to do justice to that particular moment. So I was very hesitant, but she was very insistent, and so I agreed to do it. So I have a couple of warnings, though. Normally a seminar goes 30 or 35 or 40 minutes. This lecture was actually planned at an hour, so please be prepared that this is a little bit longer than normal. It will be about an hour. So the other thing that I wanted to say about this one is that I always like to be prepared for things. And so I had this Malbridge lecture prepared a month before the ISB meeting. And the title was The Accidental Scientist. And those of you who were at the ISB, they saw that the title of my talk would be The Accidental Scientist. And then all of a sudden I realized with a couple of days to go that this lecture doesn't make any sense whatsoever. I completely threw it out of the window. The last couple of days did something different, and that's what I came up with. So there's another lecture somewhere on my computer that I thought was good and then completely rejected it, so hopefully this one will be better. So when I thought about this, it started at the Brazilian Society for Biomechanics in Florianopolis in 2015. I was chairing a workshop with 6 international people in motor control, neuroscience, and biomechanics, and then we had a roundtable discussion for about an hour. And then at the very end, and I was leading the whole discussion, at the very end I said, well, is there any last question that anybody might have? And then this woman held up her hand and she said, well, I would really like to know what is the greatest impact that the 6 of you, you know, you're all international people, what's the greatest impact you had in science? What did you do? And Jay, the person that I'm going to call Jay, I'm not going to mention them by name, but Jay gave just a very specific example in the science that he had done. And then M, the person M, said, I really haven't made an impact. So it's an international capacity, he stands in front of a crowd of people, there's about 600 people, say, well, I really don't feel I've made an impact. And there's W is me, and I completely avoided the question by saying, well, I'm really proud of my trainees, blah, blah, blah. You know, I said, you know, some of my trainees have really gone far, and I really revel in that, and I think maybe that's what it is. But I was thinking about this, you know, as Anquan mentioned, I was thinking about this greatest impact question when I prepared this particular lecture and came to the conclusion that it's really a bit presumptuous to talk about what impact you might have made. I think it's up to other people to judge what impact you might have made on your field rather than on you. But I thought instead of that, instead of thinking about what impact I might have made, I could talk about things that changed my own thinking, because that's me, and there are discoveries that we have made in the lab that clearly and profoundly changed my own thinking. And I said, I can talk about that without really offending anybody. And so as I was preparing this talk, I was thinking about really 4 discoveries that changed my thinking profoundly. And so I'm going to talk about these 4 stories here today. So I'll tell you kind of 4 related but independent stories in muscle mechanics. And I tell you a little bit the stories behind these. And so the 4 discoveries that I want to be talking about, I gave them short names. Stability of sarcomeres is one of them. Force enhancement above the plateau is another one. Passive force enhancement is another one. And then Titin force regulation is another one. And I know most of you are not really muscle mechanics, and all these things probably don't mean a thing to you, to most of you. And I hope that I'm going to be able to at least conceptually be able to convey what the importance of these stories might have been. And so I didn't want to only talk about the science, so I also wanted to talk about the stories behind them. And this story, Stability of Sarcomeres, I call On Intuition. And then the next one, I talk about truth and knowledge in science. And the next one is going to be serendipity. And finally, the last one will be about paradigms. And that's really where the title of my talk comes out. So here we are. Story number 1, stability of sarcomeres and intuition. So imagine it's the early 1980s, and you're Walter Herzog, and you're writing your thesis, your PhD dissertation, and you're writing it on the distribution problem in biomechanics, which means you try to predict individual muscle forces and ligamentous forces and bony contact forces and all that stuff. And those of you in biomechanics know this is a complex problem, and those of you in biomechanics also know that you need to know a little bit about muscles and muscle properties when you do that. But up to that time, in the sport biomechanics lab where I was, there was no muscle mechanics course, no muscle physiology. I had never done an experiment on muscles. I didn't know anything about muscles, but I needed to know how muscles worked. I needed to know their basic properties. So what do you do as a student? Well, you go into the literature and you read the classics. And one of the classics, of course, is Hill, 1953, where he talks about the descending limb of the force-length relationship and says, the system is unstable and a long continuous creep is observed on this, whatever that means, descending limb of the force-length relationship. And then you read, you know, a paper by Albert Gordon and Huxley Andrew Huxley and Fred Julian in 1966, again talking about the descending limb of the force-length relationship. And they say, well, there is little doubt that this creep that they observed on the descending limb of the force-length relationship is due to the progressive development of Sarcomere length non-uniformities and the instability represented by the negative slope. And so, okay, what's the big deal? So we have here a force-length relationship. This is just the plateau region and the descending limb. So we have this negative slope here. This is sarcomere length as a function of force. And so people were arguing that sarcomeres are unstable on this descending limb. And in fact, if you take the crossbreed theory and you model sarcomeres in series, and you see how many sarcomeres can really stay on this descending limb, then if you have a fixed end contraction, it's really only one that can stay on there. All the other ones have to go to some kind of a positive slope if you assume that this is actually the dynamic behavior of muscles, like people have done. And so really what you then would expect to happen is, that these sarcomeres are pulled apart and, are pulled onto some positive slope, as for example, illustrated very schematically here. And so that's what it is. And so I'm going, okay, good. I don't know anything about muscle mechanics, so that's what it is. And this instability was used to explain a variety of different properties of muscle mechanics. As I mentioned already with Hill and Huxley, they explain creep properties with that. They explain damage in eccentric exercise with that. They explain the force loss after eccentric exercise, and then also a property that's called residual force enhancement, a history-dependent property. So there's a bunch of properties that were based on the development of these instabilities in the development of sarcomere length non-uniformities. And so here I am, early 1980s, never having had anything about muscle mechanics and muscle physiology, and I'm looking at the entire force-length relationship on the sarcomere level as given by Gordon, Huxley, and Julian. And I'm looking at this and going, okay, so this is stable and this is unstable. And then you look at that and you go, that's a bit surprising, because about 50% of the work potential of a muscle is in this area, and this is about 60% of the working range of a sarcomere. And so you go to yourself, well, here is this universal molecular motor that's found in every animal that I'm aware of. It's always the same, it's always acting in myosin, and it really doesn't work. It tears itself apart over about 60% of its working range. And that just didn't make any sense, but of course I didn't know anything about it. Hill and Huxley are 2 Nobel Prize winners, and so you go, well, they must be right, but it really didn't sit. And then I looked a little bit, so what's the evidence? Who has ever stretched sarcomeres in series onto the descending limb and actually observed this instability? And then you go, well, nobody. There's really no direct evidence. There's this creep evidence and residual force enhancement evidence, but it's all indirect. Nobody had actually ever really made the conclusive measurements where they pulled sarcomeres onto the descending limb and looked what happened. And so there's really no direct observation of instability, at least that I could find. And as I said, this is about intuition. So I'm going to myself, well, why would a muscle be unstable over 60% of its working range? Why would muscle damage itself? Why would nature, evolution, God, whoever you believe in, come up with such a solution that I really didn't find was terribly, terribly good? And here we are, 20 years later. So now it's the early 2000s. So this was when I was a graduate student in the early 80s. This is in the early 2000s. And finally, we had the very first single myofibril setup in Canada, one of just 2 in North America, and about 1 of a handful in the world. And so you could now do experiments with sarcomeres that are arranged in series. And as you will realize very, very quickly, I only do simple experiments. I don't seem to have the brain to do anything complex. So I just thought, well, we stretch sarcomeres on the descending limb of the force-length relationship, and we just see what happens. Either they are stable and the length stays constant, or they are unstable and they indeed tear themselves apart. And there is your solution. It's a very simple experiment. And this was done by Dilson de Rossier, who was a postdoc at the time in my lab. And so this is how the experiment worked. We take a myofibril at some short lengths and activate it and then you pull the myofibril to a longer length, and now all these sarcomeres here, these are all individual sarcomeres, as a function of time, all these sarcomeres are now in the descending limb. This one is on the plateau. So this one should be about twice as strong based on actin, myosin, filament overlap than this one. So the expectation is that this one instantaneously is pulled apart and this one becomes shorter, and then I don't know exactly what happened in the middle there, but what we observed was that they actually stay all nicely the same, despite the fact that according to the crossbreed theory, this should only really have half the force compared to that one. And then you do this dozens and dozens times over again, and you find the same thing. Here we have even sarcomeres where we start on the descending limb, and then here all of these 6 or so sarcomeres are on the descending limb. And you could argue, yes, there is, there's slight movement of the sarcomeres, but there is not this popping that was predicted by the theory, or that people have predicted, that happens within 100 milliseconds and the muscle is tearing themselves apart. That's clearly not happening, because this is a time frame of about 20 or 25 seconds. So this is very, very, very long. And so really the conclusion from these experiments were then, sarcomeres appear to be perfectly stable on the descending limb of the force-length relationship. And so, you come back to the intuition. And so I think intuition is a big part in the science that you do. I think you should always believe in your intuition. You should follow your intuition. But my mechanics instructor and my co-supervisor in Iowa, James Andrews, always said, in engineering, intuition invariably leads you down the wrong way. So you always have to do math, you always have to do the experiment, and therefore never ever trust it implicitly. And so you might have an intuition in the 1980s, and 20 years later you can test it. And so don't just take it, but go back there at one point and test it. And also look for simple explanations, look for beautiful explanations. I think muscles that tear themselves apart really didn't strike me as very beautiful. And also not very simple. These theories seem to be very convoluted, and so I like this much better. And I think today now, a lot of people have done stiffness experiments and stability experiments, and I think people have come to the conclusion that that negative slope that everybody sees in the force-length relationship, of course, has nothing to do with dynamic stability and stiffness, because when you stretch a muscle, you'll get a positive slope, especially if you stretch it quickly. And so that really came out of somewhere that I'm not really sure exactly where. And then, of course, the basic thing, don't trust everything you read. So here we are, story number 2, force enhancement above the plateau, and I entitled this story about truth or knowledge in science. And before I go there, I need to tell you what force enhancement is, and then tell you what force enhancement above the plateau means. So force enhancement is this interesting property that muscles have, and they look as follows. So here we have a single impact fiber, and we keep its length constant, and we activate it, and we get a relatively steady amount of force. And now you take this single fiber, and we stretch it a little bit, and we get this little bump that we can somehow explain, and then we get an additional amount of force, and we stretch this single fiber a little bit more. You can also do it with a muscle, and you get more force. And what you have to realize here is that here you are at steady state, you are exactly at the same length, the activation of the muscle is exactly the same, and the basic theory that you read in textbooks about how muscles contract, the crossbreed theory, will tell you that in this situation the force has to be the same and you get the same amount of force. But you don't. You don't. And people have known that for longer than the crossbreed theory, but for some reason this property was not incorporated into that particular theory, and I'll talk a little bit about that later. But then the question really became, can this force, this additional force that you get after stretch, can it be higher than the plateau of the force-length relationship? Again, so this is the plateau of the force-length relationship, the isometric force-length relationship. Can my enhanced force be here, or is it always here or below that? And the reason why this is an important question was because if it is higher than the plateau, there is really not a very easy way to explain it with the crossbreed theory. However, if it's below the plateau, then you can easily explain it in some way with cross bridges and crossbreed thinking. And so really the idea then was, well, is the crossbreed theory applicable here or is it not? That really was the question. And again, as always, the experiment is essentially very, very simple. You pull sarcomeres or fibers or muscles, and you just see whether or not you get force above the plateau. And in fact, we didn't even have to do the experiment, because the truth that I'm talking about in the mid-1990s, when I was thinking about that, the truth already existed, because Paul Ekman had done exactly that experiment in 1982. He wanted to see whether or not force can be above the plateau or not. And so he stretched single fibers, intact tibialis anterior fibers, and looked, here is the plateau forces, the isometric contractions, and then he stretched and looked at the steady state forces here, here, and here, and also a little bit further down, and essentially came to the conclusion that you can approach this plateau, but you cannot exceed it. And that was what everybody believed, and I believed that, and everybody believed it. And so, okay, so this can actually be explained with the cross-bridge theory, and so we don't really have to do the experiment. So the cross-bridge theory can explain the residual force enhancement property. No questions asked. So when I started thinking about it, about 8 or 9 years later, Jenny Wakeling, who's the second author here, came to the lab. She was the wife, some of you might know, James Wakeling, who was a postdoc for Benno, and she was a veterinarian and was bored and didn't want to sit at home, and so she did some work for us in the lab. Don't tell the university, because nobody's allowed to do work anymore in a lab without going through hundreds of hoops. But in those days, you could actually just take somebody off the street and say, hey, do a couple of experiments for me. And she took intact single fibers. In fact, she developed that technique for us. She had beautiful surgical skills. So she developed that technique for us. And I asked her to stretch, get the isometric force length relationship, and then stretch these intact fibers on the descending limb of the force length relationship, and look at the steady state forces that then are shown here, here, and here, and here as well. And what we actually wanted to look at is a phenomenon that there seems to be an average stiffness when you stretch that gets steeper or stronger or higher when you're further down the descending limb of the force length relationship. And if you think of this as the average stiffness, then indeed this seems to be steeper than that, and other people have observed that as well. And I'm not going to go into details, but that was a very surprising result, and that's what we wanted to see, whether or not it was correct, and it seemed to be correct. However, what we then also saw by accident, we go, oh wow, look at this, we have some of these forces that are clearly above the plateau, on average by about 12%, and some of the individual measurements were as much as 15 or 20% above the plateau of the force length relationship. And so all of a sudden we bump into this result, and then of course did more tests, and other people did tests to kind of verify that. So it seemed that all of a sudden, from one day to the other, force enhancement above the plateau was possible, and therefore probably the cross-bridge theory cannot really explain that, or you would have to go through some very strange mathematical reorientation of the cross-bridge theory before you could explain that. And so we thought, well, there must be something else that is happening here that we cannot quite explain. And so in order to try to explain that, Tim Lenger then did this particular experiment here. And you can see here, this is March 16, at about 1:27 p.m. And this is where Tim captures a single sarcomere. So this is a myofibril silicon nitride level for force measurements. And so is going to go there, where the Z line is, and then you have the A band here and the next Z line there, and he'll try to isolate a single single sarcomere. And Tim, in 2010, was the first person ever to isolate mechanically a single sarcomere and then do mechanical testing. And here it is, you can see one of those, and he tried about hundreds and hundreds of these to get about 12 at the end of the day within about 2 years. And when you try long enough and are steady enough and have long enough patience, then you can do that. So anyway, he was able to do that, first one ever, and then did these type of experiments where he had an isometric contraction at a long length on the descending limb, where you get very little force because there's very little overlap, that's 3.6 microns. And then he stretched the sarcomere from its optimal length of about 2.4 microns. So this is the maximum force that this sarcomere can exert, considerably higher than here. And then he stretched it to exactly the same length and held it, for a very, very long time, for a very, very long time. And you can see that this force, of course, is substantially higher than here. So you get this force enhancement in a single sarcomere. But not only that, you get actually much more force than you get at optimal overlap, which is the plateau of the force length relationship. So even on a single sarcomere or a single myofibril, you get force above the plateau. And the reason why this is important is because a lot of candidates for possible achievement of this force fall away when you work in a single myofibril, because you really don't have very much left in a single myofibril. And then here are some of the results from about 6 or 8 individual sarcomeres. He did 12 in the paper. I didn't show all of them, that it didn't get too crowded, but the result was always the same, that even for the lowest one, the force was higher than the plateau of the force length relationship. And of course, then you go back and you go, well, how could this happen? And you go back to the Edmund paper, the one that showed us the truth in 1982 there, or in the 1990s, where we're thinking about that. And then, of course, we go, well, here he stretches from 2.4 microns approximately to about 2.6 or 2.7. The more he stretches, the more force he gets. And all we did is we stretched from 2.4 to over 3. All we did is we stretched a little bit more. And you can imagine when you stretch more, you seem to get more force. And he already showed that. So if Paul Edmund had just stretched a little bit more, he probably would have gotten exactly the same result. And you kind of look at that graph retroactively and go, oh my goodness, I should have seen that. It's so obvious. Once it is there, you go, he had the result already. He had it, it's right there. Just one more stretch, another 0.1 or 0.2 micron, and the result would have been exactly the same as what we had. But what did I want to tell here about the truth? Because for us and for me, all of a sudden the truth about a certain fact that I believed in changed. And I guess the point that I want to make here is that in science, truth is temporary. Truth changes all the time, and therefore you have to be able to change your thinking. I always like people to think back about 500 years ago, you know, how people and scientists thought about the world and how we think about the world right now, you know, and everything that we think about. You know, the Earth is not flat anymore. The Earth is not in the center of the universe anymore. Planets are not orbiting around the Earth in circular patterns. So everything that was thought about, or how we perceive the world and how science perceived the world 500 years ago, is really mostly not correct anymore. And so what is true today might not be true tomorrow, and in 500 years from now, everything that I'm saying, everything that you're finding, everything that's ever been found is probably not true anymore. So it's a changing world, and so you have to be able to change your thinking. You have to question what you know all the time, and you have to look at other people's results very, very carefully, and you have to look at your own results very, very carefully as well. Truth is temporary, and it goes away. So my third story is about serendipity, and it has to do with passive force enhancement, another property in skeletal muscle. And of course, you could argue that the force enhancement above the plateau was already serendipity, because we found that the experiment was not designed to do that at all. We just found that by accident afterwards. And so I could have used that one as well. And you could look at my career and say, everything I ever found that made any impact on anything was purely by accident. And this one is my favorite example, because this was not on the horizon at all. Not at all. And we thought we had made mistakes in the experiment and so on. Passive force enhancement. So here we are in the late 1990s, and we are starting to think, you know, how can you get that force? How can you get forces above the plateau? How can you explain potentially force enhancement? And people have, you know, made arguments about that there might be a passive element that might be engaged. Noble mentioned it in 1992, Paul Edmund in 1996, and then Mario Forcinito, Marcelo Epstein's student who graciously also worked with me, came up with this, you know, this beautiful engineering solution about the passive engagement of passive element that he published in the Journal of Biomechanics. And what it was he had, you know, a sarcomere or the muscle here, and then he had an element in parallel that had a little spring. So we have an elastic element in parallel. And all Mario said is, he said, well, if upon activation somehow the contractile machinery is kind of engaging here this element, then we have all of a sudden more force compared to when you just stretch it passively. When you stretch it passively, you only stretch this, but when you stretch it actively, maybe you engage something and you have to stretch this additional passive element. He modeled that, and you know what? with this very simple, very simple mechanical model, you can predict force enhancement very nicely, and actually some other properties as well. So we have been thinking about that there might be something happening passively, but nobody had any evidence for that. Zero. And so there really was nothing to go by. So here we are. This is 2002. It's not 2002. It was published in 2002, but it was probably done in 1999 or 2000. And so Tim Leonard and I do this experiment on cat soleus muscle. And the experiment was aimed at looking at the amount of force enhancement that you get at different parts of the force length relationship. The idea was you get little force enhancement on the ascending limb, maybe a little bit more on the plateau, and lots and lots and lots on the descending limb. And so we did these experiments on the descending limb at the very end, because they have passive force, and when you stretch here at very long lengths, you might actually tear apart some of the collagen fibrils, and so you might damage the muscle at these very, very long lengths. So we did this at the very end. So this is an isometric contraction at very long lengths, and that's why you have quite a bit of passive force. And then we stretch the muscle from a shorter length to a longer length, and we get about the same amount of passive force. And now we do the same. We stretch the muscle, but we activate it first. We activate it, and then we stretch it. We get the force enhancement as we expected, and then we deactivate the muscle, and all of a sudden we are here, and we are at the yellow point. So imagine here you have 3 times the same muscle at exactly the same lengths, no activation, completely passive, and all of a sudden you have more force. And some of you know the story. Rachel Shacker was sitting at the computer, and I got mad at her because I said, "Oh, you're pulling the muscle way too far." And she said, "No, I'm not." And I said, "Yes, you are, because you have way too much passive force." And then we quibbled and quabbled until I sat down on the computer and I did it, and I sent her to look after the muscle, and then realized, no, it was right. And then we did it over and over again and tested and retested again. And indeed, when you do this stretching at a long length, you get this additional passive force in a muscle. And so we labeled that passive force enhancement that never ever existed before 2002. And now there's many laboratories around the world that look at these properties and try to understand it and try to know where it's coming from. And we are one of these groups. And we tested this passive force enhancement, made bigger stretches, and we got more passive force enhancement. We showed that it was very, very long lasting, but also showed that when you shorten the muscle here in the green trace, if you shorten it all the way, that the passive force drops to 0, you can immediately unlock it. So that passive force enhancement persists, but if you shorten the muscle just briefly and then stretch it back up again, it can instantaneously be gone. It's like it's unhooking, it's like a mechanical unzipping or so. And so we are trying to figure out what is happening here. Anyway, we kind of tested this. We showed that this passive force enhancement is correlated to the entire force enhancement. And so we tried to publish this. And the Journal of Experimental Biology was fabulous at the time, because, usually after one round of revisions, you're rejected. But this one, I think, went through about 3 or 4 rounds of revisions, and they actually allowed us over and over again to resubmit it. And so we resubmitted it. And then at the very end, all the reviewers were happy. But then one reviewer said, if you just stretch the muscle actively, you get more passive force. Surely, millions of people, well, not millions, but hundreds of people have stretched muscles. Somebody must have seen that. You cannot be the first ones. And I'm going to myself, yeah, the reviewer is right. Somebody must have seen that. So I went back into the literature, and sure enough, many people had seen that. And the one that I like the best is David Morgan, who published a paper on exactly the same muscle preparation as we did, cat soleus, 2 years earlier. And here we go. So here, David Morgan, he has an isometric contraction here at this length, and then he stretches the muscle, actively at different speeds, and shows very, very nicely that the force enhancement is speed independent. You get exactly the same force enhancement, deactivates, and has a couple of seconds here of deactivation, and very clearly this force is higher than that one. And the way it looks, probably would stay a little bit higher for a long time. And of course, this is about 2/3 of that. But didn't make a remark on it. And several other people have exactly the same results. So we were not really the first people to see it, but we were the first people to say, hey, what's happening here? This is exactly the same length in a passive muscle. We didn't damage it because people thought we had damaged it, but no, we got exactly the same active force. When we deactivated the muscle, put it to a short length, we got exactly the same passive force as we had previously. So there didn't seem to be any damage or anything. And so we're going, well, what's happening here? Well, as I mentioned, this is my serendipity story. So we we found this and we looked at this, and this was absolutely not planned. We were not looking for that. We had no clue what we were doing, really. We knew what we were doing, but we were looking for something completely differently. And so it's purely by accident. However, I would like to give myself a tiny bit of credit, because my mind was prepared. And what do I mean by that? Tim knows that, and other people have done experiments with me know that. Sometimes you do 600, 800, 1,000 tests with capsuleus, because it's a muscle that you can stimulate and stimulate and stimulate for hours and hours and hours. With Tony Guimaraes, we did 36 hours of experiments with capsuleus. It doesn't get tired. You get tired before the muscle gets tired. You can test it forever and ever and ever. So you do hundreds and hundreds of experiments. But the way I go into the experiment is that each single trial is written down beforehand in my notebook. So at 4 o'clock in the morning, when I'm completely dizzy and can't think anymore, I know trial 751 is this one. And trial 752 is this one. And not only that, I always make little schematics drawing, forced time drawings of what I'm expecting to happen. And of course, when I looked at my little schematic drawings there early in the morning, I realized, no, the passive force should be exactly the same. So something is wrong here. And so although it was a serendipitous finding, I can claim at least that I had an open mind and a prepared mind, and I'm going, wow, something is wrong here, and we need to have that. And so I think it's always important when you do experiments to have a framework in mind, that you kind of know what you are expecting to happen, even though it might not happen at all. But I think it's important to have that. And then, of course, when you find something that completely doesn't make any sense, then you have to test and retest and so on to really satisfy yourself that it's all happening. So I'm coming to my last story here, Titin force regulation, and I want to talk a little bit about paradigms. And those of you who know me, they know that Titin is kind of a little bit my favorite topic. So this is going to take a couple of minutes longer than the other stories. But I want to talk a bit about paradigms. And so, what is a paradigm? Thomas Kuhn, in his book, The Structure of Scientific Revolution, shows very nicely, gives a framework what paradigms are, and it's a philosophical book, and I'm not a philosopher, so I kind of translate it for my own. And on my own, I would say, paradigm is a generally accepted theory in science that explains most observations. Very, very rarely do paradigms encompass everything and can explain everything. And paradigms can be tested. You can set up hypotheses, and then I also believe in hypothesis testing very, very much in the sense of Karl Popper in 1934, who talked about the falsifiability of hypotheses. The whole idea, not only is truth changing, but you actually never really know scientific truth. You cannot really ever accept a truth. You can only support it with more and more and more evidence. And of course, the famous example that people always give about the falsifiability is the whole idea that you have this hypothesis that you go around the world and you see all these white sheep, and so you make the hypothesis that all sheep in the world are white, but you can never ever prove that, because you cannot in a millisecond see all the sheep of the entire world simultaneously, and therefore ascertain absolutely uniquely that right now at this instant in time all sheep are white. So you came to the experiment. You can look at more and more sheep and travel the world and so on. You can support more and more evidence, but you never really know if you're true. However, you can falsify the hypothesis very easily. You go to Scotland on vacation, you see that one black sheep, and you go, whoop, no, not all sheep are white. So it's easy to reject a hypothesis, but it's very hard or impossible, Popper would argue. And I think in the natural sciences, in the natural world, I'm definitely a believer of that you can never ever prove anything to be true. So not only is true not really ever true, but it's just kind of supported by a lot of evidence to the best of your knowledge. It changes continuously as well. So we are really strange people that we work in this make-believe world where we tell ourselves what's correct and what's not, and we really actually don't know anything at all. And then of course, these paradigms, every once in a while they'll shift. There is what Thomas Kuhn will call a scientific revolution. So in 1954, in muscle mechanics, there was a scientific revolution, because before the 1950s, people thought that muscle contraction and force production was done by the shortening of the myosin filaments. So the myosin filaments would somehow magically fold up and shorten and produce force. And then the two Huxleys, Andrew and Hugh, came along. They said, no, 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 you have two sets of filaments that slide relative to one another. And then all of a sudden, from not from one day to another, but over a decade, people started to think about muscle contraction in a very different way. We have actin, we have myosin, they slide relative to one another. Then, of course, Andrew Huxley in 1957, the famous article, where he then said not only that, but that sliding is accomplished by these cross bridges that are attached to the myosin filament, and they cyclically interact with the actin filament and then pull the actin past the myosin filament, as shown here. This is the thicker myosin filament, this is the actin, these are the cross bridge heads. There's an elastic tether there, and here you have an ATP hydrolysis that then powers this particular event, and that's how we think about it. Force is produced by actin and myosin in muscle, and that's it. So again, I was going that was about 2008 or 2009, and I don't remember exactly when, and I don't even remember where I was, but I was invited to give a seminar at some university, and I talked about the cross-bridge theory and force enhancement and passive force enhancement. And then again, at the very end, people always seem to ask the best question at the very, very end. The last question that this woman asked me was, so when you do these myofibril experiments, so how far can you actually stretch a sarcomere before it breaks? And I'm going, I have no idea. I have absolutely no idea. So I'm going home and I'm going to Tim and saying, we need to pull sarcomeres, we need to pull myofibrils and just see how they break apart. And so Tim, of course, nice person he is, did that. So he stretched these myofibrils passively, and I only show it to 6 microns because they fail at about 6.5 to 7.5 microns, and then force would drop to 0. But I'm not going to show that because it's not important where they fail. What's more important is that I told Tim, well, we know that muscles have a different mode of failing when you stretch them passively compared to actively. And therefore I said, well, stretch them actively as well and see what happens. And of course, it's very easy to do. You activate the myofibrils and then you stretch them. And then, you get more force here. Of course, you get more force because you have not only the passive force, but you have also the actin and myosin interaction. But then the problem came here when you stretch them more than 4 microns, because here you start losing overlap between the actin and myosin filaments. And therefore, there's no interaction anymore. There's no active force anymore. And therefore, the passive force, of course, as everybody knows, will drop down here and then follow the passive trail because we only have passive force. Everybody knows that. It's easy. However, that's not what happened. The force climbed and climbed and climbed, and at the end here at 6 microns was about, on average, whatever that is, 700 units, whereas here it's about 200, so it's about 3.5 times bigger or so. So we're going, what's happening here? There's no actin myosin anymore. And so, well, the only thing in a myofibril that really can exert passive force is Titin. That's why it's so beautiful to work with myofibrils, because there's nothing there anymore. All the collagen is gone, everything's gone. But Titin, the structural element in the sarcomere, is still there. And so we said, well, let's eliminate Titin from these and stretch actively and passively. And as we expected, when you eliminate Titin, you don't get any force, neither actively nor passively. There's nothing, complete instability. That's when you have instability. There's nothing that comes out of the muscle. So we figured, well, this possibly could be Titin. But then a very clever reviewer said, well, I want you to be sure that you don't have somehow still cross bridges attached in some manner, despite the fact that you're a very, very long lengths. And so we did additional experiments where we stretched these sarcomeres beyond overlap. So we made sure that all of them are above 4 microns. And then we stretched them there, and then we let them relax for 10 or 20 seconds. And then here we deactivated the muscle. So we deactivated the myofibril, and if you still have actin and myosin, then of course this force would drop down somehow, but it didn't. So we put the relaxing solution in there, it stayed there, suggesting to us that no, there is no remnant actin myosin, there's no cross bridges, therefore Titin has to be the culprit and has to be, responsible for that. And now most of you sit here and they say, Titin, what the heck is he talking about? I've seen the movie Titanic, but I'm not really sure what Titin is. So Titin is the biggest protein that has ever been found, and it occurs essentially anywhere, but it occurs in skeletal muscle in a very distinct manner. It starts here in the middle of the sarcomere, is rigidly attached to the green myosin filament, runs freely here on the I band, and then attaches here into the Z line, and also in the last 50 to 100 nanometers also attaches to actin. So it forms a bridge between the actin and the myosin that is there. And in this area, you can pull it apart and you get force, the passive force, and then when you shorten the muscle, then it kind of folds up again. And so it's been called a molecular spring because you can extend it. And so we're going, okay, so we need more force in a myofibril. We don't have actin and myosin anymore. The only thing we really have is titin. So somehow this titin might become stiffer, and therefore when we pull the muscle, it might get stronger and produce more force. And so our speculation was that you have a titin here, and you take a muscle and you pull it passively, and you get some passive force from titin in a myofibril or in a single sarcomere. And then the idea was, well, if you activate a muscle, and activating, of course, means that you throw calcium into the contractile machinery, we thought maybe that calcium doesn't only bind to troponin C and allows for cross-bridge binding, maybe the calcium also goes to titin, binds in certain regions, and maybe reinforces some of these molecular bonds and makes it stiffer. And so we said, well, let's test that. So we take single myofibrils and we stretch them in different calcium concentrations at the same time as we inhibited cross-bridge attachment by troponin C. So we took the troponin C out and then tested that. And these experiments were done by Venus, who is sitting right there. And so she did these experiments with a low calcium concentration. When you stretch the muscle or the myofibril, you get less force, and when you have higher calcium concentration, you get more force. And remember, actin and myosin are thrown out, so this is just a force. So we thought this must be an increased stiffness in titin, and therefore we get more force. And so then we look a little bit at the molecular makeup of this titin, and we are working with this one, the rabbit psoas, myofibril mostly. And so when you look at this, you see a lot of these rectangles, these brown rectangles, and these brown rectangles represent, each one of them represents an immunoglobulin domain. So we're going, okay, maybe calcium binds to these immunoglobulin domains, and maybe when calcium binds to these immunoglobulin domains, it becomes stiffer and therefore makes the muscle stronger when it's active compared to when it's passive. And so we did that experiment. So this was Mike Duval, who received a plasmid from Jane Clark from Cambridge so that we could reproduce recombinantly immunoglobulin domains. And then we used fluorescent spectroscopy to see whether or not calcium was binding to these immunoglobulin domains. And we found that yes, indeed it does. In fact, it did in a dose-dependent manner. The more calcium you threw in, the more binding that was happening. And when you took the calcium out, then it was unbinding. And then we took, and then we looked at this atomic force microscopy, or I have to say Mike Duval, looked with my atomic force microscopy whether or not it was really harder in the presence of calcium to pull these immunoglobulin domains apart. And here we have the control condition with resting levels of calcium, and here is the force as a function of the unfolding of these individual peaks of the immunoglobulin domains. And here it's in the presence of calcium, and this is about 220 here, this is about 180, so it's about 20% more force on average that is required to pull these immunoglobulin domains apart. So we were really happy. We're going, oh wow, calcium binds, it changes the mechanical properties of titin, we are home free, we can explain how it all works. And other people had then shown the same. So Siegfried Labeit showed very, very nicely that calcium can bind to this E-rich or glutamate-rich region of another area in titin that's called PVK. And then Venus showed the same. And then Mike Duval was the one that I just showed. He showed that calcium can bind to the immunoglobulin domains. Today we know that there's a bunch of things that can happen to titin to change its stiffness and therefore its mechanical behavior. Not only calcium binding, there's also phosphorylation, there's disulfide bonding, there's a whole bunch of things that have been shown in the meantime that will change the mechanical properties of titin. And everybody in this area fully believes that that's happening. You can change the properties of titin, no questions asked. The only problem was that this calcium binding explained only a teeny tiny amount of the results that Tim found. In fact, it only explained about 10, if you are very, very generous, it explained about 20% of the increase in force, but then we are very, very generous. More realistically, probably about 10%. So we are still missing 90% of the force from our experiments and other experiments that people have done. And so I'm going, well, another way of how you can make this spring stiffer is by eliminating part of the spring from elongating. And again, I'm thinking in very simple terms. So I thought, well, Titin is already attached here in the last 50 nanometers or so to actin. Maybe it zips up somehow upon activation and attaches rigidly to the actin filament, so it cannot elongate here anymore. So it becomes a very short spring that then, of course, would be much stiffer if you stretch it the same amount. And so it would be rigidly attached here to the actin as it is always to the myosin filament. And so we're going, okay, let's test that. And so we thought, for a variety of reasons, that we knew where this might be binding and where this area, what this point might be, where it might be binding. We thought it was the so-called PVK region. And so we put an antibody very specifically there, and then we labeled that antibody with a fluorescent tag so that we could see how much the proximal segment of Titin, how much the distal segment elongates. Again, very, very simple experiment. You just put a marker on there, and then you measure how this elongates and how this elongates. When it passively elongates, you would assume that the proximal and distal segment elongate. And very simple experiment, when you actively, you assume that the proximal segment does not elongate, and all the elongation is taken up by the distal segment. Very simple. Took Mike Duval about 3 years to get the techniques all going, but after 3 years, he had these beautifully lighted myofibrils where you see these fluorescent tags attached to these so-called PVK areas. And now we could do these experiments where we pull it passively and actively, and all we do is we measure the lengths of the proximal and distal segment. So here we are. This is the Titin segment length. This is the sarcomere length. And so this is the half sarcomere. So we take this half sarcomere and we stretch it from 1 micron to 2 microns, or a full sarcomere from 2 to 4 microns. And we measure how this elongation changes. So this is the proximal segment. And as I said, when we passively stretch, we would expect this segment to get longer, longer, and longer. And to nobody's surprise, this is actually happening. So we stretch, and this is the actual data from one myofibril, but we have about 130 of these, and they all look about like that. You stretch passively, and this thing gets longer, longer, and longer. And so here it's almost 1 micron at about 4 microns of sarcomere length. So this happened. And so now we do the active stretching, and in the active stretching, the idea is this binds to actin, and it doesn't elongate, and therefore all the elongation is taken up here. However, to our big disappointment, that really never seemed to be happening. But on average, what seemed to be happening is that we stretch a little bit, and we get a very similar elongation as we had in passive condition, and then all of a sudden, it didn't stretch anymore. And so we're going, oh, something strange is happening here, and we don't really know exactly what might be happening here. But one of the explanations, my favorite explanation, but I have absolutely no clue if it's correct, my favorite explanation is that it somehow might attach to actin here. So once more, if I show the whole animation, it goes, goes, goes. After a little stretch, it somehow gets stuck there, and then all the elongation has to be taken up by the distal segment. All we really know from these experiments is that the Titin elongation in the active and passive states are very different. And those of you who have seen Mike talk about it, he thinks about this very differently than I do. So we look exactly at the same data. He interprets it different than I do. And so, we are the same, we are the same lab, the same everything. But in his thesis defense, he didn't defend it that way. He had a different theory about what might be happening. But both theories involve that somehow part of Titin gets entangled, gets stuck, cannot elongate, and therefore all of a sudden you have a shorter spring, and therefore you get more force. So the conclusion here is that Titin appears to bind to actin, thereby shortening its free spring length. Titin appears to play a crucial role in muscle contraction, so it doesn't seem to be only actin and myosin. We are convinced, and a lot of more people are now convinced, that there is other structural proteins that likely play a role in active force production when a muscle is activated. Exactly how that works is unclear. And so really the new paradigm that we are suggesting is as follows. So here you have the active force length relationship between actin and myosin. So this is the plateau and the descending limb of actin and myosin. And then here this is passive force that is partly due to titin, but also to collagen and other structural proteins that come into play. And all we are suggesting is that this is true, this is true for an active muscle only between actin and myosin, and this is true for a passive muscle only. But what we are suggesting is that when you activate a muscle, this passive force does two things. It becomes steeper because titin is stiffening, and it starts occurring at a shorter sarcomere length because you reduce the slack length of one prominent structural protein that produces passive force, and that is titin. And so we think that in an active muscle, the structural proteins produce much, much more passive force than they do in a passive stretch. And therefore, of course, when you measure active force and you measure passive force and you subtract the two, then you subtract this passive force rather than that one. And my hunch is that that's probably completely wrong when you then try to estimate the active force really that's based on actin and myosin. But as I said, there's a lot of work that needs to be done to really figure this out. So in 1999, Andrew Huxley was at the ISB meeting, in a muscle meeting that they organized in the mountains. Here he is with my two boys at the time at the Drumheller Museum. That's when my kids were still little. They are very tall now. In fact, I'm missing, I want to make a point of this, I'm missing my son's thesis defense right now as I'm presenting. He's defending his master's degree, looking at C. elegans and cell division under circumstances that I don't understand at all. There's a quote there. This is Huxley's little book, Reflections on Muscles. And I want to read the entire last paragraph when he talks about eccentric contraction. So what Huxley says, this is in 1980. This is about 4 years after titin has been discovered, and really nobody had any clue about the function of titin. But Huxley says, elongation of active muscle is something that happens in very many of the movements that animals make. And we all agree with that. And I imagine that special features have been evolved which allow this elongation to take place without damaging the muscle. Then it goes on saying, I suspect that many of the unexplained phenomena, such as those I have just described, and he talked about force enhancement and passive force enhancement and all kinds of things, so where am I? So all these special features I have described just here are related to these special features and have little relation to the processes that take place during shortening contractions. There's a wide and probably difficult field for investigation here, and I expect that it holds a number of surprises. Andrew Huxley, 1980, in my view, he predicted titin. A special feature to me is titin that really explains a lot of what might be happening here. So what did I want to say here in this last one about paradigms? Well, when you work in paradigms never really explain all observations. Be very careful about that. New paradigms, what I always find interesting is I think that new paradigms, and that's something that you as young scientists, the students here need to know, new paradigms before the crossbreed theory will be replaced and you add titin in there. You need to prove that this new paradigm is much, much better than the old one, explains much, much more, not a little bit more, but much, much more, and contains all the predictions that were done correctly previously. So you cannot really all of a sudden throw something out. And that is, science is very, very conservative in that aspect, that you really have to go beyond normal proof when you want to change a paradigm, because you invest in printing new textbooks and shaping the thinking of the new people and changing the thinking of all the people in the field, and therefore you need to have a lot of proof. And I always say, science therefore has to be conservative. And I always tell people, you have to think about that when you publish manuscripts, because people and scientists, 99.9% of the scientists think that the peer review process is there to eliminate bad papers. I don't think that's correct, because if a bad paper gets published, it really doesn't matter. It really doesn't matter very much, and we know there's open access journals, you can publish anything, but nobody will read it anyway. So I think the main purpose of the peer review process is to make sure that the good stuff, the paradigm shifting stuff, is very well explained and thought out, and all the experiments have been done, and otherwise you reject it. You don't let it in until it's there. And therefore, in a review process, the very bad papers typically get rejected, but the very, very good papers get rejected as well. And you have to think about that. All the people in my lab that work on myofibrils, they know how hard it is to publish that stuff. The easiest things to get published are summer student projects. You work in a paradigm, you have that one teeny tiny question that's exactly in the paradigm. The summer student comes, you ask the simple question, the simple experiment is done, you write it up, you filled that little teeny tiny hole that exists in the area, published, everybody's really happy. But when you start doing stuff that might potentially be, working on paradigm, it's very, very hard to do that. And think about that. So next time a paper of yours get rejected, look at it very carefully and think, is it really bad or is it really good? And hopefully, coming out of this lab, of course, it will be really good and paradigm shifting and so. But I think it's something to keep in mind, because there's very famous examples where papers have been rejected by nature. And the one that I know personally is Krebs, with the Krebs cycle that was rejected by nature. And he had this, thing, the rejection letter in there, and he was mad at nature. And I'm going, no, you should be happy that nature rejected it, because it showed at the end of the day it was a good idea, and it made you go back to the lab and do more experiment. And at the end of the day, you won the Nobel Prize for it, but only after it was rejected, and you did due diligence. And I think that's necessary. And the reason why it's necessary is because there's a lot of people, there's a lot of ideas, and some of them are just not that good. And if you let all those ideas into the literature, it would be very, very confusing, I think, for a new person to potentially come in and really figure out what's right or wrong. Something to think about. So what are my reflections here? Well, the simplest is usually the right solution, I find, in the natural sciences. The correct solutions are usually beautiful, and I find Titin adaptation a beautiful solution. If it works that way, it would be so simple, so beautiful, so nice. I have no clue if it works that way. And I don't care if at the end of my career people say, oh my God, Herzog was full of shit and nothing was done correct. But at least I have proposed something that a lot of laboratories are now looking at very, very seriously and try to test whether or not it works or not. It would be a beautiful solution. It doesn't mean it's the right solution, but something might be correct there. So these are my very final thoughts. I talked in the first story about intuition. Some of you might have taken the Myers-Briggs test, and supposedly if you're an INTJ personality, that makes you a good scientist. N stands for intuition, T stands for thinking. Those are the two important attributes that you need to have. Needless to say, I'm not an N, I'm an S, and I'm not a T. Believe it or not, I'm an F. For those of you who have taken the Myers-Briggs test, they know what that means. They know what that means. That means I don't make any rational decisions ever in my life because I'm always driven by my feelings rather than my thinking. I shouldn't be a scientist. The most prominent person that always comes up under my personality profile is Mother Teresa. I am Mother Teresa. I have the personality profile of Mother Teresa. Whereas here, here you have Isaac Newton and all the big guys, and so you should be that. If you're me, you're never going to make it in science, except of course if serendipity throws you some bones, which it did to me. So that's what I want to say here. Think about the fact that truth in science is a very changeable thing. And as I said, in the natural sciences, in muscle contraction, it's just an example, but you never really know exactly what is true. We can build up support for certain things, but then at the end of the day, they might actually turn out to be incorrect. And truth will be changing, and I guarantee you in 100 years from now, the world will be looking very, very different. So keep that in mind, which that allows you also to not really take yourself and your research and your results to be taken too seriously. I talked about serendipity, but have a prepared mind. When serendipity strikes you, be prepared to take advantage of it. Paradigms in biomechanics, the field that I know, there's really not very many paradigms in the true sense that I'm aware of. I've been very, very lucky to work in a field like muscle mechanics where there is a paradigm, there is something that is in the textbooks and everybody essentially accepts it and all students learn it. And so you have something to orientate yourself, and I think that is very, very good. My last slide, this is, I'm going to change, Tim Leonard is in the back there, and he's already getting red and embarrassed because I gave a long talk about this last slide here, because behind every scientist there is a Tim Leonard, and I said a lot of things at the ISB, but I'm not going to say a lot of things. But the one thing, just this afternoon as I was thinking about this particular slide, Al Baines, who some of you might know, a colleague of mine and researcher, about 20 or 25 years ago, when we were both young, he was young and I was young, he told me, I was always having ideas and this type of thing. And Al Baines said something that I thought was really interesting. He told me once, because I said, I have this idea and this idea and this idea. And Al Baines said, if you can think it, you can do it. If you can think it, you can do it. And I thought that was a really nice thing. If you can think it, you can do it. Except in my case, I have to slightly adapt it. If I can think it, Tim can do it. And always has. Thank you very much. Thank you.
Talk from Walter. I'm sure you have lots of questions. The floor is open. I'll ask the first question. The last slide, the bottom right corner, there's an H-O-N-H. What does it mean?
That's for you to figure out. That's for you to figure out.
It just shows you the usefulness of psychological tools. But anyways,
I was asking people who had seen that in the original talk, and I don't think anybody had.
Well, first of all, Walter, I want to thank you for doing this. I know I made you do it in a sense, but I had heard that the biomechanics presentation was great in Australia, so I really thank you for doing that.
Thank you.
And I think you, hopefully for everybody in this room, you inspire us with your curiosity, and I hope that's what we're doing with all the students we have in kinesiology in all our areas, to be curious about what you want to research. So thank you for doing that. I do have a question though. In the passive force enhancement piece, when you said that researchers didn't see it, it was there in their research, but they didn't see it. Do you have any thoughts on why they didn't see it?
I think once, people who do this type of experiments that stretch muscle, they are typically interested in the work that's performed during activation, and so everything is interesting when the muscle is active. And then once you deactivate it, the experiment is over. And in many, many of the experiments, you actually don't see the deactivation and how the force drops to zero. Most people cut it off, but then several people did not, and those that did not, you actually see it. But I think the experiment is over as soon as you deactivate the muscle, and that's why nobody pays attention to it, or paid attention to it. And the only reason why we paid attention to it is we had to be very, very careful how far we stretch the muscle, and that we are exactly the same lengths as for the reference contraction. And then we had the increase in passive force. I'm going, oh no, Rachel, you pulled the muscle too long. We are at the wrong lengths. Until we then realized, no, it's actually not an error in the experiment. This is actually what happens.
So you can just argue against your supervisor sometimes, right? Get someplace. Can you just leave the door open though before we all dine in here?
It's very hot, isn't it?
Yeah. Right.
Just maybe leave the door open, yeah. Yeah, great, thank you.
I want to follow up to that and then ask the question. So I think another reason why people might not have seen it is that force measurement is hard, and you don't often trust your force measurement for a long period of time, especially if something changed, because you expect if it's lots of different kinds of force measurements, or if they're just not reliable, and it happens. So my question is, you started with this force creep thing where force was steadily going down and people were talking about Sarcomere's popping. So what does explain that?
Well, that's actually a very interesting question, because and I always, actually, when I talk in my 663 class, my muscle mechanics class about that, when we go onto the descending limb and we have a fixed end contraction in single fibers or in myofibrils, we don't see that force creep. And people who have done what I call force clamped experiment, like the Huxley Albert Gordon, Huxley and Julian there, they did not take the fiber and hold it fixed at its end. But what they did is they put into the middle of the fiber, about 5 millimeters apart, they took 2 markers, and then they tried to keep those 2 markers at the same length during a contraction so that that middle segment stayed isometric. And then they adjusted that by shortening or stretching the fiber at the end. So if this thing became shorter, they would stretch it so that it would stay the same length. If it became longer, they would shorten it. And we have tried to do experiments where we do a very similar thing, where very slowly we change the lengths in fibers. And as soon as you do that, force goes up or down. And so in that particular experiment, my hunch is, but I have no proof for that, that maybe the middle segment was slowly trying to shorten, and that they stretched the fiber slowly so that you get an increase in force there. In our intact fibers, I don't see that. I'm going on the descending limb. On the myofibrils, we are on the descending limb. I don't see that creep where force goes up, and up.
I have a question about Mike's results, where you see the kink in the length of the proximal G domain. If it is catching, why do we not see a similar kink in the force record when it's being stretched?
I think part of the reason might be because I would assume this is not all happening at the same time. We think that in the myofibril we have about 2,700 titans. And when we look at the 130 experiments that Mike does, then sometimes we find that in certain sarcomeres at a given length, at 2.1 microns, at 2.3 microns, at 2.5 microns. So we see it occurring at all kinds of different lengths. And my hunch is that in a given titan or in a given sarcomere even, that this does not happen all at the same time, that you stretch all the titans and then all of a sudden they all stop there. So I think that is not happening.
Okay, so the kink would happen at different sarcomere
lengths. If we could observe each individual titan, which we cannot, there's no way to do that at the moment, if we curve through that, and if it really entangles the way I'm suggesting, then I would assume that happens kind of stochastically somehow along the way as you're stretching. That would be my hunch.
But it occurs at the same point in each sarcomere, which would suggest that there's communication between titin molecules.
It could potentially occur at the same point in each sarcomere. The thing, of course, is that we have a band of markings there with the fluorescence, and that's relatively wide, and we take the centroid of that. So we are really getting an average measure of the average behavior of all the titans. And I wouldn't want to bet a lot of money that the individual titans do the same thing, or maybe that they are synchronized in a given sarcomere. But we do know when we look at sarcomere per sarcomere per sarcomere in a myofibril, then it doesn't occur at the same lengths of the myofibril in each of the sarcomeres.
What about the width of the signal? Would that increase with the active stretch compared to the passive stretch?
The width of which signal?
The width of the fluorescence signal. Because if it's binding at different locations along the actin, you'd expect that the width of the signal should expand as well.
As far as I know, I mean, I've seen visual records of this, and visually it's not apparent, so it would not be a big effect, but we have never really very carefully looked at whether or not in the passive and the active, if you measure kind of the edges, you've made an edge detection algorithm, whether or not you would actually get wider, and that's actually very good. That's actually a very good suggestion. Maybe you could go back to some of those results and do that. Yeah, because that would be a logical explanation. If what I'm saying would be correct, then you would expect that, yes. Yeah.
So I like your explanation on titan boost regulation.
Yeah.
I think it is simple and beautiful. I'm curious about what's the fundamental benefit of having titan working like that in biological systems.
Well, so the question really was, what's the fundamental benefit if Titin really worked that way? And I talk mostly about force enhancement, but the fundamental benefit would be that if you have a spring there that gets stiffer upon activation, then that would stabilize the myosin in the middle of the sarcomere. And also, since the force goes quite dramatically up there on the descending limb, when you lose force in the actin-myosin filament overlap, then you get the result that Tim observed, that force even on the descending limb goes up, 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 and up. And so you have a positive slope there, and you get an inherent stability of the sarcomeres that are serially arranged. So, centering the myosin filament, giving stability to the sarcomeres on the descending limb. Because if you use the cross-bridge theory just with actin and myosin and its stochastic interaction, you get instability. The cross-bridge theory in that particular situation would predict that the system is unstable. So it would provide that stability, and then it would provide an explanation for the force enhancement, the passive force enhancement, and also something that Venus Juma showed is that in this force enhanced state, and I wasn't even talking about that, in this force enhanced state, you need much less energy, metabolic energy, ATP, for a given amount of force than you use in a regular isometric contraction. So after a stretch, you get more force for less energy consumption. And of course, that would be possible if you have a passive element that somehow engages, because that doesn't really take any energy. And there is a Titin analog in some invertebrate muscles. It's called Titin. It has a very similar structure to Titin, and it does exactly that. Titin is known to make a bridge, like the cross bridges between actin and myosin, and then it can hold force in these particular muscles. So for example, clam shells, I guess, do that you can support a certain amount of force in the activated muscle there with this Titin, then bridges a force between actin and myosin, and it essentially doesn't cost any energy at all and produces a certain amount of force, quite a substantial amount of force supposedly. But that's really the literature, that's not experiments that we have done. But there is muscles that are known with this Titin mechanism to produce long forces for hours and hours with virtually no energy cost.
So if the bridge is there, it will stay there without costing too much energy more?
In that particular instance, in the clams and invertebrate muscles where that has been found, yes, the bridge stays. And in fact, the on-off state occurs not with calcium or anything, it occurs with phosphorylation. In the dephosphorylated state, it's on, and in the phosphorylated state, it's off and detaches. So phosphorylation regulates the Titin state. Yeah.
Just one more.
Sure.
Does it cost energy to build that bridge?
I'm sorry?
Does it cost energy to build that bridge?
Well, whenever two proteins combine, either the combination of two proteins or the rupture of two proteins cost energy. You cannot have it without energy both ways. But of course, if you only attach once, and let's say that would cost energy, let's say 1 ATP, then that would really not be very bad, because if you have to cycle cross bridges, because cross bridges cycle about 15, 20, 25 times per second under normal conditions, and every time it cycles, you need an ATP. So when you exert a certain amount of force with a muscle, you're using a lot of ATP continuously. Whereas when you attach a structural element, you would use ATP once, either if it attaches or detaches, but then it would stay there, and then it would essentially be energy free. It's like you just put a spring between these two things and push them together. It takes energy to attach the spring, but once the spring is there, it would exert force forever and ever until you take it away.
How long is the delay in between when you have the passive force enhancement and then you turn the muscle off and then you turn it back on again and you still have the same enhanced force afterwards? Is it very short, or can it be extended to these kind of longer periods of time?
So the passive force enhancement in cat soleus muscle, it actually doesn't stay there forever. When we measure it and we have a lot of passive force enhancement, then we can see that the force slowly but surely decays. And it takes, on average, we measured it once in several muscles, it takes about 3.5 minutes before it goes away. So the passive force enhancement is not there forever and ever and ever, but it stays in cat soleus for about 3.5 minutes.
And is that then just the process of pumping out the calcium out of the...
I don't think it's calcium, because calcium would be gone much faster than that. And also, I don't think it's calcium because when we then, we hold it there for 5 or 10 seconds, and then we just relax the muscle to a short length and stretch it right back, it's immediately gone. So I don't think it's calcium regulated. I think there's another regulation, and that regulation can be taken away mechanically by shortening and stretching the muscle back, which I find very intriguing, and we've never really looked more carefully at that, but at one point we need to.
Yes.
So at some point you said that the active force that you measure is not only active, but active plus passive. So do you believe that when we activate the muscle, there is in the beginning this rate of force, of increase of force that we see, is also a part of Titin attachment, especially at long runs?
I don't know that. The idea is when you activate a muscle, might part of the force that comes in directly be Titin attachment, that some titans might attach and you might get more force, and that even in an isometric state, it might not all be actin-myosin. I have no idea. The results from Mike would suggest maybe no, maybe you need a little bit stretch before you get that. But I'm really not sure.
I have one question.
Yes.
So I'm here more of asking for advice. So you talk about the Sarcomere instabilities. When you first come across that in 1980, something like that, it took you 20 years before you can actually start stretching the Sarcomere instead of descending. So within that span of time, how do you keep your level of motivation high so that you can achieve the goal?
Yeah.
Years ago.
I don't think.
How far away?
I don't think there was a level of motivation. It's just a result that I inherently, from the very, very beginning when I read that, it just never really appealed to me. And, and with Dr. Epstein, very early on, we were looking at instability. And we had once a visitor, Truskinovsky, Lev Truskinovsky, who came and spent a week with us many, many years ago, exactly talking about those issues. And then he came up with different models. So it's always been there in my mind somehow, and then we did also instability experiments and tests with Todd Allinger, and have a paper together again with Marcelo Epstein. So the thinking was always there, but until we could do what I felt was the deciding experiment. You pull it out there, they're all at the different lengths, they're all on the descending limb, they should tear themselves apart, but they don't. To me, that was kind of the important experiment to do. But we tried over the years doing other things, but it took us a while to get. You always have to imagine that my PhD thesis was purely theoretical, and I had not done a single experiment on muscle when I came to Calgary. And the reason why I came to Calgary is because I wanted to do, muscle experiments. I came to Calgary, as some of you know, not because of the Human Performance Lab, not because of Ben O'Neill, not because of anything. I came here because of Andy Hoffer, because I wanted to validate my individual muscle force prediction that I had in my theoretical model with direct measurements of muscle forces in a model. And Andy Hoffer had this beautiful model where he could measure the electroneurographic, the electromyographic activities and the muscle forces, multiple muscle forces. And so I learned these techniques from him. We had these cats walk back and forth and jump and run and climb trees and whatever, and we predicted what the individual muscle forces would be with the model that I had. And then, of course, the end of the story is that my model was completely wrong, was not accurate at all. I never ever should have gotten a PhD. It was essentially useless. And but to my satisfaction, I don't think there's a model out there to date where anybody would say, whatever you do with your arms and hands and whatever you do, I can always predict muscle force. And I'm not accurate, but I'm always plus minus 10% of the absolute force. Nobody in the world can claim that today. And so nobody has solved the problem even 30 years later. So maybe I deserve my PhD because I didn't make a contribution. It was useless, but nobody has solved the problem in the meantime anyway. So maybe it's a bigger problem than I thought it was.
Last question for Walter. Now let's thank you.
Thanks a lot.
Right, next week, Dr. Salvatore Federico, sitting here, will close the HBO seminar series of the fall term in 2017 by telling us the story of becoming a biomechanist by chance through an adventure in continuum biomechanics. See you next week.